I'm Lauren.
And today we are going to be talking about proteins. So we all know that inside every single one of our cells, we have DNA. But how come, Lauren, if I have hair and I have my skin and I have nails, if all of them contain the same DNA, why do they all look different?
I don't know.
That's really just going to be because we have proteins that are going to be coding for different things that will make our hair, it'll make our eyes. And so proteins are super important for us to live. It creates differentiation between all the species in the world. Proteins are really what guide life on Earth. In order to do this experiment, we are going to need 4 different transparent cups, and those are just going to be used as different trials, things like that. We're also going to need 1/3 a cup of rubbing alcohol, as well as 2/3 a cup of water. We're also going to need an egg, a microwavable container, as well as a fork and a pair of scissors. You also can use some sticky notes and a pen if you want to make sure you're labeling things and staying on track. But first, before we begin any experiment, we need to make sure that we are dressed correctly. So the very first thing we're going to do to start this experiment is label our 4 cups, and we're just going to do that with some sticky notes. The very first one is going to be labeled our control. Our second one is going to be labeled tap water. Our third one is going to be labeled rubbing alcohol. And then our fourth one is going to be labeled hot water. Now we're going to be putting in the contents into our respective containers. So with our control, we're not going to be putting anything in there because it's our control. We want to see what goes on with the experiment. The second one, tap water, we are going to put in 1/3 a cup of tap water. Next, we're going to add in 1/3 of a cup of our rubbing alcohol into its container. And lastly, we are going to be adding in another 1/3 a cup of, this time, hot water to our last container. So we're just going to add in some of that hot water to our microwavable container until it's boiling. So we are just now going to separate our egg yolk from our egg white while our water is boiling, and so we just grabbed 2 extra bowls so that way we can do this. So now that we have our egg white separated from our egg yolk, we are going to take a pair of scissors and we're going to try to cut it into 4 separate parts, and each part will go into one of our containers. Now we're going to be pouring our hot water into our hot water container. Now that you've poured the hot water into the container, we are going to start a timer, and we are going to put 1/4 of each egg white into the container. So if the time is going on and you still don't really see much happening, you can use your fork to mix them up to start the process a little bit faster. Is completely up, we are going to be looking at these and making observations. So Lauren, can you tell me what does it look like in the hot water container?
I see some white stringy things.
Yeah, we see some white stringy things. What about our rubbing alcohol?
I also see white stringy things, and it's a little bit cloudy.
Ooh, and it's cloudy. What about our tap water?
There's a little bit of white stringy stuff.
A little bit, and then our control?
Nothing happened.
Yeah, there's nothing in our control. And this is because we didn't add anything to our control. If you leave an egg white out, you're not going to expect anything to happen. Now that we've made our observations about what's happened in these containers, we can start to make predictions. So we know that the protein in our control, it stayed the same since we couldn't observe any differences with it. But all of our 3 other containers did have differences. We saw this white stringy stuff. And that's because for our tap water, if we think about what's in our tap water, we sometimes have different metals that are in it. It really just depends on what's in your pipes or what's in the city's water. Now, if you were to use filtered water for this experiment, I want you to think, would there be the white stringy stuff that shows the denatured protein, or would it look more like our control? So moving on to our rubbing alcohol container, this one is very white and stringy. This one, the protein denatured a whole bunch. And we can predict why that happened. We know that our proteins in our bodies are in very specific pH solutions, and if we change that by adding alcohol or an acid or some type of base, then our proteins are going to unravel. Now let's think about the last one with the hot water container. If we add in some kind of thing that changed the temperature, this would also break down our proteins. And this is why when we have fevers, it's so important for us to make sure that our fevers go away, because it could ruin the proteins inside of our bodies. But proteins are not only in eggs. Like I said before, proteins are found in our bodies. They're found in any living thing. And so this doesn't just stay true for eggs, but think about protein denaturation across all organisms or all cells.
Hey Hope, is that why we have to cook eggs?
Good question. What do you guys think? Thanks for watching today. See you later, scientists.